This is our biosensor presentation. How it works. The plasmids have a biosensor called red fluorescent protein, RFP, that reacts to light. When the pDawn is exposed to light, it activates the RFP gene, and the bacteria will be a bright pinkish or a red color. When pDusk is in the absence of light, it will trigger the RFP gene and turn the bacteria a pink color. When pDawn is placed in the dark, it will be a cream or opaque color. When pDusk is in the presence of light, it will become opaque. This was our experimental design. First step, we made the agar. Then we added 15 microliters of kanamycin. We poured the 7 plates of the mixture and labeled them. We have 2 control samples, 1 pDusk and 1 pDawn. 5 experimental samples will be compared to the control samples. Our control will be light intensity. Our variable will be the tinfoil used to cover the plates. We will put our pDawn control sample closest to the light bulb, while the 2 other will be a further away distance from the bulb. The pDusk control will be covered completely. 1 pDusk will be covered on the top and bottom, and the sides will be open. 1 will have the sides covered and the top and bottom open. The half pDusk, half pDawn plate will be covered on the pDusk side and uncovered on the pDawn side. Our hypothesis was that the pDawn closest to the light will be the most pigmented. Then as the samples get further away, they will be less pigmented and turn more opaque. The pDusk sample completely covered will be the brightest pigmented. The sample with the top and bottom covered, but the sides open, will have a less bright pigment overall, but will be brighter in the middle and more opaque on the outer edges. The sample with only the sides covered and the top and bottom open will have small amounts of pigment on the outer/inside edges closest to the tinfoil. The half and half sample will be brightly pigmented on both sides. These are our light intensity graphs. On the left, they go by 0.0, 0.2, 0.4, and 0.6, so you can see that that's a higher number. The one on the right is within a tenth of a point, so that is the lesser intense graph, so that would be the bottom. The one on the left would be the top. Our conclusion is that the light intensity is a factor on the pigmentation shown in pDawn. When we placed the pDawn plates on a mechanical crank table, which resulted in greater light intensity than the plates that were placed at the bottom of the box. The pDawn plate that was closer to the light was more pigmented than the plate that was on the bottom of the box. On the other side of the experiment, pDusk, when it was completely covered and not exposed to light, was a faint pink. When exposed to light, such as only covering the top or only covering the sides, it was fairly opaque in color and showed a more creamish tone. This proved our hypothesis because it showed light intensity is a factor of pigmentation. What we would do differently. We would repeat the experiment to confirm results, maybe more than once if we had to. We would do a better transformation of pDawn and pDusk in the first trial and write labels on the bottoms of the plate, not the top. We would also use less experimental samples so we can keep our experiment organized.